What is biodiversity? Biodiversity generally refers to the enormous variety of life on Earth. To understand biodiversity more clearly, we can say that this term is used to describe every living thing on Earth, from humans to animals to plants, down to the tiniest of microorganisms. We value biodiversity for many reasons. Biodiversity is important both for what it provides to humans, such as food, shelter, and water, and in its own right, such as helping us understand the relationships between different species and their interactions in their environment. Biodiversity is an extensive field and is divided into three types: ecological diversity, species diversity, and genetic diversity. So ecological diversity refers to the variety of ecosystems within a particular locality. This type is also referred to as ecosystem diversity since it involves the complex network of several different organisms living together in a given environment and the interactions between them. Next is the species diversity. This type of diversity refers to the variety of species within a habitat or a region. Species diversity can also be defined as the number of different species present in an ecosystem and the relative abundance of each of those species. Lastly, we have genetic diversity. Genes are the basic units of all life on Earth. It's responsible for both the similarities and the differences between organisms. Now, genetic diversity refers to the variety of genes within a species. This means a species may have different populations, with each having different genetic compositions. At its simplest, biotechnology is technology based on biology. But what exactly does biotechnology do? Biotechnology uses, harnesses, and deliberately manipulates biological systems for the efficient manufacture or processing of useful products. It is a multidisciplinary science that moves through various fields such as medicine, agriculture, environment, and industrial fields. Like the stripes of a rainbow, different biotechnology applications are grouped in different colors which represent different research or development areas. The first one we're going to discuss is green biotechnology. So green biotechnology or agrobiotechnology is the application of biotechnological tools and techniques to genetically improve organisms and crops for the betterment of the population. It holds promise in producing crops with high yields and nutritional content, insect resistance, extreme weather resistance, and longer shelf life. With green biotechnology, scientists have developed solutions to increased agricultural productivity, enhance breeders' ability to make improvements in crops and livestock, and enabled improvements that are impossible with traditional interbreeding. Scientists also aim to achieve, with the help of green biotechnology, the removal of toxic heavy metals from the environment with plants, convert plants to biofuels, and transform waste to natural sources of energy.
Where biotechnology is used for biotechnology in the medical and pharmaceutical industry and health preservations. This branch involves the productions of vaccines and antibiotics of generative therapies, creations of artificial organ and new diagnostic of disease. The importance of weird biotechnology has become a very important part of the field of diagnostic, gene therapy, and clinical research and trials. So, white biotechnology is solely applied to the industry to replace polluting technology for clean ones. The technology uses living cells from woods, bacteria, plants, and yeast to synthesize the easily degradable products. Blue biotechnology is biotechnology using aquatic organisms. Think blue for the water that these organisms live in. But blue biotechnology goes beyond this, utilizing an incredible variety of organisms from earth, oceans, lakes, river, and stream for many different purposes. Importance of blue biotechnology plays a major role in aquaculture or fish farming. High quality fish foods, disease resistance, species diversifications, and even environmental impact are all aspects of aquaculture that involve blue biotechnology.
Genetic engineering, technique that allows genes and DNA to be transferred from one source to another, leading to the production of living modified organisms, LMO, or genetically modified organisms, GMO. is called as genetic modification or genetic manipulation. It is a direct manipulation of an organism's genes using biotechnology. These are the pros and cons of genetic engineering. First is the pros. Tackling and defeating disease, getting rid of an illness in young and born children, potential to live longer, produce new food, organisms can be tailor-made, faster growth rate in animals and plants, pest and disease resistance. The next one is the cons. It may lead to genetic disease, limit genetic diversity, reduce nutritional value, risky pathogens, and negative side effects.
What are genetically modified organisms? A genetically modified organism is an animal, plant, or microbe whose DNA has been altered using genetic engineering techniques. Some GMO animals, however, are produced for human consumption. How are GMOs made? GMOs are created by selecting and inverting a particular gene into a plant to produce a desired trait such as resistance to pesticide. Farmers have been selectively cultivating plants for thousands of years. Creating genetically modified organisms are more scientific process. Possible application for the genetic modification of organisms. Some benefit of genetic engineering in agriculture are increased crop yields, reduced cost for food or drug production, reduced need for pesticide, enhanced nutrient composition and food quality, resistant to pests and disease, greater food security.
We all know that genetically modified organisms have been artificially manipulated through genetic engineering to express new traits. There are a number of possible applications for genetic modifications of organisms, and there are also examples of that. For the examples, we have here BT corn, Aqua Advantage salmon, golden rice, Savers tomatoes or Saviar tomatoes, and glow-in-the-dark animals. For corn, it is modified with the bacterial insecticide gene so that it produces insect toxins within its cells, protecting it from pest species. And we can also see the second picture, the salmon, which modified with the growth hormone regulating genes in order to grow to market sizes in significantly less time. Third one, the rice, modified with daffodil genes to have more beta carotene, which the body converts to vitamin A. We have here tomatoes when modified by the removal of genes responsible for softening of fruit, which means the tomatoes spoil more slowly. Lastly, animals modified with genes for fluorescent proteins will grow in the dark, and this novel feature serves no practical purpose. So genetically modified organisms is very likely you are eating foods and food products that are made with ingredients that come from GMO crops, and many GMO crops are used to make ingredients. And if there's an example, we have also here benefits of GMO. First, we have the higher efficiency in farming, increase in harvest, control in fertility, increase in food processing, improvement of desirable characteristics, and lastly, sixth one is the nutritional and pharmaceutical enhancement. So the benefits using GMO or genetically modified organisms include crops having higher harvest yields, which can feed more people in the world and making food more nutritional. The risk of using GMOs include both the unknown long-term risk of eating genetically modified organisms crops and the negative effects that they can have on an environment. So for the next slide, we have here a question: should we be worried about GMO? So as what I've said earlier, the risk of using genetically modified organisms include both a known long-term risk of eating GMO crops and the negative effects that they have on the environment. We have here potential risk of GMO. First is inadequate studies on the effects of GMO to humans and the environment. it is a potential risk, especially to consumers, because it might harm our health while consuming genetic engineered products, and also a threat to the environment. And it only happens if the safety aspects of genetic modified foods of plant origin does not apply or is not applied. Second, it promotes mutation in organisms, which the long-term effects are still unknown. A new study shows that Monsanto's genetically modified corn and Roundup herbicide cause negative health effects in rats and is raising questions about the safety of genetically modified organisms. Some researchers also are having research on the effects of GMO to human and rats.
Hi everyone, my name is Mary Antoinette National, and I will be continuing the discussion on the potential risks of GMO. According to some studies, one of the potential risks of GMO is that it promotes mutation in organisms, and the long term effects that this mutation carries are still unknown. There have been plenty of research conducted to study the effects of GMOs to human and to rats, and some of the results include number one, premature death, and number two, some rats develop large tumors and organ damage, which includes kidney and liver. Another set of potential risks of GMO include the occurrence of more allergic reactions, gene mutation, antibiotic resistance, and a change in nutritional value. Some people believe that GMO food can trigger more allergic reactions because they contain genes from an allergen. However, it is important to note that according to the Food and Drug Administration and the Graduate School of Arts and Sciences at Harvard, there is no evidence that GMOs are any more or less allergenic than their non modified counterparts. Some researchers also believe that eating GMO food can contribute to the development of cancer. However, according to the article released by the Food and Drug Administration, GMO crops are not changed in ways that would increase the risk of cancer for the humans or animals that eat them. An analysis of data by the National Academies of Sciences, Engineering, and Medicine found patterns that reveal that cancer rates are not connected with eating GMOs.
Biosafety on genetically modified organism. Number 1. Codex Alimentarius Commission or Codex, responsible for the development of standards, codes of practices, guidelines, and recommendations on food safety, or has developed principles for the human health risk analysis of GM food product. Number 2. Cartagena Protocol on Biosafety, International Environment Treaty that regulates the transboundary movements of LMOs. Number 3. International Trade Agreement on Labeling of GM Food and Food Products, an agreement that requires exporters of GM food and food products to label their products and give the rights to importing parties to reject or accept the GM products, or consumers have the right to know the freedom to choose. Introduction to Biosafety. According to the Center of Disease Control and Prevention, or CDC, biosafety is the application of safety precautions that reduce a laboratorial's risk of exposure to a potentially infectious materials and limit contamination of the work environment and ultimately the community. Why biosafety? Biosafety is used to protect from harmful incidents. Many laboratories handling biohazards employ an ongoing risk management assessment and enforcement process for biosafety. Failures to follow such protocols can lead to increased risk of exposure to biohazards or pathogens.
Needs of biosafety. Biosafety is very important because biosafety is a framework that describes the use of specific practices, training, safety, equipment, and specially designed buildings to protect the worker community and environment from an accidental or unintentional release, infectious agents, and toxins. Ecology. Ecology is the study of the distribution and abundance of organisms, the interaction between organisms and their environment, and structure and function of ecosystem. Ecology helps us understand how the world works, provides useful evidence on the interdependence between people and the natural world, and as well consequence of human activity on the environment. Agriculture. The concept of biosafety in agriculture, which includes animal husbandry, fisheries, and forestry, entails examining and monitoring the consequence of possible gene flow, competitiveness, and effects on other species as well as possible adverse effects to products on animal and human health. Biosafety policy decisions may have long-term ramifications for agriculture sustainability and food security. Medicines. Medicines are chemical or compounds that are used to treat, prevent, or cure disease as well as relieve symptoms and aid the diagnosis of illness. Exobiology. Exobiology, also known as astrobiology, aims to detect presence or evidence of possible extraterrestrial life on celestial bodies in our solar system and beyond. Chemistry. Chemistry is the study of matter which defined as everything with mass and occupies space, as well the changes the matter may go through when exposed to various settings and situations. Chemistry aims to understand not only properties of matter such as the mass or composition of a chemical element but also how and why matter changes, whether something transforms after combining with another substance, freezes after being left in a freezer for 2 weeks, or changes color after being exposed to much sunlight. Biosafety in academic research. Biosafety also assists you in comprehending and implementing risk management practices that are specific to your facility. You are responsible of determining the optimal course of action while limiting the incidents and unexpected difficulties with risk management. Risk management is in fact a proactive approach that ensures proper containment.
Next topic is about biosafety issues. So it refers to the procedures, policies, and principles to be adopted to safeguard the environment and the human population. It refers to the containment principles, strategies, and practices that are adopted to prevent exposure to pathogens and toxins. Examples are laboratory safety. It requires great care and constant vigilance. Blood-borne pathogens are infectious microorganisms in human blood that can cause disease in humans. Recombinant DNA. It uses enzymes to cut and paste together DNA sequences of interest. Biological waste disposal. Infectious substances and diagnostic specimen shipping. Specimens may be shipped by registered airmail. The basic triple packaging system is used with the same requirement as for other means of conveyance. Another issue is respiratory protection. It may be a face piece, helmet, hood, suit, or a mouthpiece respirator with nose clamp. Next is bioterrorism and select agents. It determined to have the potential to pose a severe threat to public health and safety. Next is mold and indoor air quality. Often leads to poor indoor air quality, airborne contamination, and allergens affecting the health of everyone in the building. Next is occupational safety and health in the use of research animals. And lastly, biohazards used in animals models. Biosafety in microbiological and biomedical laboratories, or the BMBL, has served as the cornerstone of biosafety practice in the United States. The core principles of this document is protocol-driven risk assessment. It is not possible for a single document to identify all of the possible combinations of risks and mitigations feasible in biomedical and clinical laboratories. The BMBL should be used as a tool in the assessment and proposed mitigation steps in biomedical and clinical laboratories.
Principles of biosafety. Practice and procedure. The use of good microbiological techniques is the most important element of containment. Personnel working with biological agents must be aware of hazards and must be trained to safely handle and dispose of these materials. along with the standard practices and special practices and consideration. Safety equipment. Safety equipment includes biological safety equipment, safety stethoscopes, and other engineered controls designed to minimize exposure to biological agents. Safety equipment is the most effective at minimizing exposure when workers are trained on the proper use of such equipment and the equipment is regularly inspected and maintained. Facility design and construction. Facility design features include physical separation of laboratories from public access, especially designed ventilation systems and autoclaves. These design features protect personnel working outside the immediate laboratory as well as the outside of environment. Increasing levels of protection or biosafety levels are used to identify the protective measures needed in a laboratory settings to protect workers and the environment and the public. Biosafety levels 1 to 4. Biosafety level 1. Agents not known to cause disease. Biosafety level 2. Agents associated with human disease. Biosafety level 3. Indigenous exotic agents associated with human disease and with potential for aerosol transmission. Biosafety level 4. Dangerous and exotic agents of life-threatening nature. Increasing levels of employee and environmental protection, as the 4 biosafety levels also appropriately describe activities involving infectious disease work with experimental mammals. Guidelines for working safely in research and medical laboratory facility. Laboratory biosafety describes the application of specific practices, safety equipment, and special designed laboratories to create a safe environment, both within and outside laboratory for work conducted with infectious agents and toxins. Animal biosafety levels 1 to 4. Animal biosafety level 1, defined organism and not known to cause disease in healthy adults. Animal biosafety level 2, moderate risk agent present disease of varying severity. Biosafety level 3, indigenous or exotic agents, aerosol transmission, serious and potential lethal infection. Animal biosafety level 4, dangerous or exotic high-risk agents, aerosol transmission, serious and potentially lethal infection. Laboratory animal facilities, which accommodates exquisitely controlled environments for the care and maintenance of experimental mammals. Animal model that support research like fruit flies, nematode, mice, zebrafish, and there are few others like rats that are often used as well. And guidelines for working safety in animal research facilities. Biosafety concepts, the BMBL. Standard microbiological practices. Standard microbiological practices are generally defined as the basic hygiene practices that apply to all labs that manipulate microorganisms or any biological materials that contain microorganisms. Most important concept and strict adherence. Aware of potential hazard. Biological hazards includes viruses, bacteria, fungi, prions, and biologically derived toxins. They are also trained and proficient in techniques. Supervisors are responsible for appropriate laboratory facilities, personnel and training, special practices and precaution, and occupational health programs that aims to prevent and control occupational disease, work-related diseases, and injuries among workers in all occupations.
Safety equipment. Primary containment barrier minimize exposure to hazard, prevent contact or contain aerosols, engineering controls or equipment, personal protective equipment or PPE, gloves, gowns, respirator, face shield, boot, biological safety cabinets, covered or ventilated animal cage systems. Lastly, facility design and construction. Secondary or barrier engineering controls contributes to worker protection, protects outside the laboratory, environment and neighborhood. Example, building and lab designs, ventilations, autoclaves, cage wash facilities, and etc.